Today, I will be talking about the sensory neuron. Sensory neurons are a type of nerve cells in your body that carry information from your senses like touch, temperature, and pain to your brain. Sensory neurons include various types that transmit different kinds of information. The sensory system processes environmental input and quickly relays information to the central nervous system through peripheral nerves and receptors. Sensory neurons are part of the somatosensory neural pathways and have a cell body, axon, and dendrites. Dendrites receive sensory input and transmit signals through the axon to the cell body. Structure. Peripheral nerves are made up of nerve fibers supporting tissues and blood supply. Sensory neurons, which are part of pathways that help us feel things, have a simple structure with a cell body, axon, and dendrites. Dendrites receive input and send signals through the axon to the cell body. The cell bodies of these neurons are found in the sensory ganglia, either in the back part of the spinal cord or along the cranial nerves. Different types of receptors, like those for temperature, touch, pain, light, and chemicals, create electrical signals when they sense something. Functional significance. Proprioceptors help us sense our body's position using special A alpha and A beta fibers. Mechanoreceptors using A beta and A delta fibers deal with sensations related to touch and pressure. Nociceptors and thermoceptors using A delta and C fiber fibers handle pain, temperature, and such. Cranial nerves with general visceral afferent fibers, such as glossopharyngeal and vagus nerves, play roles in phenomena like brain freezes. So, this made me wonder, how can studying insect bites help us understand which nerves make us itch? And the study I used was the peripheral and central mechanisms of itch. And I found that insect bites enhance our understanding of the specific sensory neurons involved in itching by providing insight into the location, axonal structure, and the diversity of itch sensory neurons. These neurons, primarily situated in the dorsal root ganglia and trigeminal ganglia, possess pseudo-unipolar axons that bifurcate. This unique axonal structure allows them to extend both to the peripheral dermatome and the spinal cord for central transmission. Furthermore, studying insect bites helps unravel the diversity of DRG neurons, considering factors such as cell size, myelination degree, electrophysiological properties, peripheral innervation, central projection, and gene expression profiles. This comprehensive exploration contributes to a clearer comprehension of how specific sensory neurons respond to itch-inducing stimuli, like insect bites. This study used a technique called transcriptome profiling to figure out which neurons cause itching. It found three types of neurons, NP1, NP2, NP3, likely linked to itch. This study also looked at their genes, revealing receptors for various substances like histamine and serotonin. This detailed information helps us understand how itching works at a molecular level. The detailed molecular insight gained from this study paved the way for future research into the broader field of sensory neurons, function, and potential therapeutic interventions. And here are my references.